Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to perform footprint analysis in ATAC-seq data. So what is footprint analysis? Simply speaking, footprint analysis identifies transcription factor binding sites in the genome. You might be confused. Isn't this the goal of ChIP-seq data analysis? You're absolutely right. Both analyses can achieve the same goal. However, there's a big difference between the two analyses. Let's refresh our memory about ChIP-seq and ATAC-seq. What does ChIP-seq do? ChIP-seq identifies binding sites for a specific protein in the genome, while ATAC-seq identifies open chromatin regions in the genome. See the difference? In ATAC-seq data, we have no idea what proteins bind to where in the genome. So footprint analysis is specifically designed to solve this problem, to identify transcription factors and their associated binding sites in the genome. Compared to ChIP-seq, footprint analysis offers a couple of advantages. First, it works on multiple transcription factors. In fact, you can apply it to all the transcription factors at the same time if you're willing to. Second, it has higher resolution. By mapping binding motifs to the peaks, we can directly observe the binding dynamics in the motif. See the indent over here? That's where the transcription factor binds to. It's like a footprint on the grass, and that is exactly where the name comes from. Why is that? The binding protects the region being sequenced. That's why. It has less signal. If you have multiple conditions, you can also perform differential binding analysis to compare the binding dynamics between conditions. The tool we're going to use for footprint analysis is a command line tool called TOBIAS. To install this tool on a Linux system, open your terminal, directly download this tool from GitHub using git clone. Go to the folder you just downloaded and create a new conda environment using the provided YAML setup file. Once set up, you can activate the TOBIAS environment. Install max2 if you want to recall your peaks, and install Europa to annotate the peaks. This is another way to install packages in conda environment. Pip install, and you can check the TOBIAS version. Next, we download the transcription factor binding profiles from JASPAR database. Using wget, you can directly download the entire database or the individual transcription factor binding profiles, depending on if you know what you're doing. The downloaded database is in this format. So this is the name of the transcription factor, and this is a matrix. It's called a position frequency matrix. The numbers represent the counts for each position, and this matrix represents the binding motifs. If you don't understand, don't worry about it. This file is not for you to read. It's for the TOBIAS tool. Next, let's prepare our input data for TOBIAS. The code here looks complicated, but don't get scared yet. TOBIAS only requires two input files, a BAM file and a peak file. In our case, we have two groups, Group A and Group B, and we have replicates in each group. So the code here is just to create a folder for BAM files and peak files for each group, and move all the replicates in their corresponding folders, and merge the BAM files and peak files for the replicates. That's all it does. It's not complicated at all. So first, we create the folders for each group for the BAM and peak files. And next, we move the replicate BAM files and peak files into the corresponding folders. And next, we merge the band files for each group. And next, we merge the peak files in each group for the replicates. If you don't have peak files, don't worry. You just need to merge the band files and recall the peak for the merged band files using max2. Once we have the input file prepared, we can start running TOBIAS pipeline. Our first step is to correct the ATAC-seq data using TOBIAS ATAC-correct command line. Our input is the merged bind file, the merged peak file, our genome file, the blacklist file from our previous tutorial. The output is the corrected ATAC-seq data. We want to use 16 CPUs. So what does this step do? Well, you know, TN5 doesn't cut the genome randomly. It has bias. So this step corrects the bias to make the indent more prominent. Next, let's calculate the footprint scores using TOBIAS footprint scores command. Input is a corrected bigwig file from our previous step, and our merged peak file. Output is a bigwig file containing footprinting scores. We use 16 CPUs. We do the same for both groups. And this is how we calculate the footprinting scores. Basically, it's the flanking region signals minus the mid-region signals. The resulted bigwig files can be directly visualized in IGV or can be used in the subsequent analysis. Next, let's prepare the input file for the differential binding analysis. Again, the code looks extremely complicated. However, the logic behind this code is super simple. Because the differential binding analysis only requires one peak file, which is the combined peak file from group A and group B. All this code is just combine the two peak files and format the combined files. Let me explain it to you. First, let's combine the peak files from group A and group B and store the combined file. Next, we download the GTF, the genome annotation file. Then, we annotate the combined peak file using Europa. Next, we format our combined annotated peak file. And then we get a header, which is the column names for the combined annotated peak file. And then the last step is to remove all non-standard chromosome names from the combined peak file. And that's it. Now we can perform our differential binding analysis using TOBIAS bind detect command. The input, the motifs, are from our JASPAR database. You can use the entire JASPAR database, or you can use individual transcription factor profiles. Here we use the entire database. Signals, group A, the bigwig file that contains the footprint scores from group A and also group B. Genome is from our human genome. Peaks is our combined, merged, annotated, formatted peak file from our previous step, and also the column name for the peak file. Alt directory is the group A versus group B, which contains multiple files that you can explore. You can rename the group A and group B using 16 CPUs in this step. The results contain multiple plots and the table, the different table in this format. It also contains a volcano plot in the HTML or PDF format. Lastly, we can directly visualize the binding dynamics of a specific transcription factor in group A and group B using TOBIAS plot aggregate. The input is the binding sites of a specific transcription factor we identified in group A and group B. Here we use MyLG as an example, as well as the footprint scores for group A and group B from our previous step. The output is this plot. You can also smooth the curves in the plots using the smooth option. In this figure, in the first column shows the binding scores for the binding sites specifically identified in group A. In the middle column, it shows the binding scores for the binding sites specifically identified in group B. And this is the overlap of these two plots, and this is the overlap of these two plots. And this is the overlap of these two plots, and this is the overlap of these two plots. And the last one compares the binding scores for all the binding sites identified in group A and group B. You should use this one. This is all I have for today's tutorial. Subscribe if you like it, and I will see you in the next video.